Hey, good afternoon, everyone. My name is Tess Baker, and I represent Valdosta State University. Tuberculosis. While I'm sure most of you didn't blink an eye at that word, for 2.3 billion, or 1 in 3 people worldwide, this word is still considered a death sentence. Pictured above is an exchange student who attended an American university for months until she was finally diagnosed with multi-drug resistant, or MDR-TB. A disease that can spread by a single sneeze was essentially right at our back door. To understand the international problem and learn more about a needs for a solution, we met with foreign embassies from several countries with high TB burdens to discuss drug development and collaboration. And we learned in December of 2015 that the White House issued a national action plan for MDR-TB. This is a worldwide problem with a corresponding market. Antibiotic resistance in TB has grown so severely in over the past decade that the medical community isn't so worried about active TB as it is the 4 different resistant strains of TB. In 2012, bedaquiline was the first MDR-TB antibiotic to gain FDA approval in over 40 years. However, the average cost for a 6-month treatment with bedaquiline is around $250,000 US, and the side effects from it are horrific. New antibiotic developments average around $5 billion and contain lengthy, uncertain results. So, instead of creating stronger, harsher antibiotics, why not make the old ones work again? We have developed a solution to overcome antibiotic resistance in tuberculosis. Our solution is a complex that revives existing frontline TB antibiotics. These complexes have undergone clinical trials at NIH against both resistant and non-resistant strains of tuberculosis. And we have filed patent application and peer-reviewed articles on our data. Our complex, again, uses existing frontline TB antibiotics. So think of our approach like a Trojan horse. The antibiotic is hidden inside the complex with no definitive size or shape and is made unrecognizable to the bacterium. The TB bacteria then consume the unrecognizable complex encompassing the antibiotic and die. All 4 components are crucial to impact the TB bacteria. Without all 4 components, it does not work. Antibiotic resistance isn't going away. The stronger we make antibiotics, the stronger the bacteria will become. We have a sustainable solution to antibiotic resistance. Thank you.
Nice job. Good job. Judges.
I have a couple questions for you. PEG is not new in the sense of delivery.
No.
What was your MIC levels and the difference between those resistant strains and the normal strains of TB?
We have on our 5th slide.
Can we have graphics, please?
This is kind of like our poster data from NIH when they sent us back our resistant trials. And you can see the first 4 are showing just different species within the complex by itself. Like INH against these 2 resistant strains, MIC values were well over 200. But the more you added in each component, by the bottom you had MIC values within killing range of the 2 different resistant strains that they had.
And you have done this also, I guess, with Ramson.
Yes, ma'am. It's been done with caprimycin, amikacin, isoniazid, and rifampicin.
All right. What's the level of copper that you use, if I may ask?
The level of copper, I don't have the specific numbers on it. I can get that to you, though, afterwards.
Just wondered about, because copper, too high levels in the body will eventually sometimes induce different types of cancer. But so you have no side effects. This has not been done in human trials. You've only done in vitro?
It's been in vitro. We did just get our ADMET data back, though.
Okay.
And it was well below the toxicity level, so that was really promising. And residence time was still really good. But we're thinking about not doing a pill so much as more so a nebulizer type.
Oh, okay.
So it's more direct, and you wouldn't have that long-lasting residents with
And you
copper.
don't, if it's just going to be sold overseas, so you don't need FDA approval in this country for delivery systems?
Ideally, yes, we are in collaboration with South Africa. There's a research institute that their embassy put us in contact directly, and they're really pushing this.
So presumably this could be applicable to other diseases besides tuberculosis. Has it been tested or anything?
We have been doing this against the 60 cell line panel of cancer for cancer trials at NCI. This same type of hidden complex has been used, and it's been getting better every single time with it. We went after TB because it is number 2 to HIV in infectious disease. So we knew it would have a lot of publicity with it, and we had really good results so far with it.
Go ahead, then on.
Okay, what's the relative cost of this compared to the standard antibiotic?
So the standard INH treatment is very affordable. In countries in like South Africa, if you have just the normal or the regular case of TB, they put you on that treatment, and the TB donor programs have made, and the countries have made that affordable in treatment. This would be very comparable because all components are easy. All components of it are FDA approved and easily, readily accessible. There would be no crazy stimulation of new species for this complex.
So one final question for me. A lot of the resistance is plasmid mediated, as well as other venues, as you know, with the microbial world, which always amazes me that it is the invisible power, so to speak, and the intimate stranger. But your patents, is it mode of action, mechanism? Because eventually this is kind of cool research to be done to unravel why is it blocking any more mutations, or it's just, the plasmid doesn't recognize it.
Right. So for that, we'll get kind of lengthy, but for right now, the patent is encompassing the component, the delivery mechanism, and the fact that it is a hidden case type delivery. We're still going back and forth with the South African Research Institute on what that mechanism of action exactly is going on. We do know that because of all the different species in it, it is causing different cellular mechanisms with the sugar and accelerating growth rate, that kind of thing. So we're still looking at that, though. But we do have some of those covered in the patent.
Tess, thank you very much. Great job.
Thank you.